So now let's move on to what types of media are out there. Something that obviously is very important. Again, we're going to use different media types based upon what our needs are, what types of organisms we want to grow, what are the optimal requirements of those organisms. So let's talk about some of these different media types and some applications of when they may or may not be used. And we'll start with complex media, which incorporate probably the majority of media types on Earth, or that are used in the laboratory, I should say. So what makes a medium complex? Well, it's all about the ingredients, and more specifically, at least one ingredient in the recipe that in which there is no exact chemical formula. So, based upon these examples here, nutrient broth or nutrient agar, which is probably the most prevalently used medium in the laboratory. If you look at the ingredients, you'll find something called yeast extract, which is basically a bunch of dead yeast cells. And the point here is that yeast cells do not have a specific chemical formula. We can't take one yeast cell and say, okay, this yeast cell is C12H14O5. Yeast cells can differ from cell to cell, so there's no exact chemical formula. Moving on, if we think about blood agar, the ingredient that is the blood is also variable. We can't say blood is a mixture of a variety of different things, different proteins, liquid, water, antibodies, and we can't say that blood has a specific chemical formula. In terms of chocolate agar, it doesn't actually have chocolate in it, but it has something called beef extract, and beef extract, again, is highly variable. So, what's the good about complex media? Well, when we look at some of these really organically rich substances that are in these examples, yeast extract, blood, beef extract, they contain pretty much any type of food source, energy source, that a microbe would want. So we don't have to exactly know what an organism likes to eat, because if one of these ingredients is in the medium, then there's got to be something there. There's probably something there that the organism will find good to eat. So that's a good thing. If we don't want to know exactly how to grow up an organism, we throw it on one of these upon one of these media types, and it probably will grow because it'll find something it likes to eat. So what's the problem? Why don't we always use this? Well, it goes back to these chemical formulas. And if you can imagine a scenario in which a group of scientists in Europe have discovered or have isolated a brand new disease-causing microbe, and they report in their results they grew it up on a complex medium. So then, due to good scientific method, many other scientists around the world want to try to replicate this experiment. They want to also try to isolate this organism from samples. So they use the same complex medium that the European group used, and they find they can't isolate the organism. Now, based upon those results and the fact that you use complex media, you can come up with two potential explanations for this. Number one, maybe the organism just wasn't in the sample. So if it's not there, then it's not going to grow. However, you could also propose that the complex media you used may be slightly different than the medium that was used in Europe. Because again, you can't replicate exactly the types, the media itself. So maybe if you use blood agar in Europe and the blood agar that was used in America was slightly different, then perhaps that slight difference was enough to inhibit the growth of that microbe, even if it was there on the sample. Now, the opposite of complex media is going to be defined media. So this is a scenario in which we know the exact chemical formula of everything placed in that mixture. So, for instance, if there's salt, sodium chloride, in the mix, or if there's glucose in the mix, C6H12O6. We know these chemical formulas. So, what are the pros and the cons here? Well, it's basically the opposite of what we just talked about with complex media types. The pro is going to be that replication can happen. You can truly replicate the medium because you know the exact chemical formula. Glucose doesn't change when you go from Europe to the U.S. Salt does not change when you go from Europe to the U.S. So that's good. But the bad part is, again, because you are really refining the types of compounds in that medium, there's probably going to be less food types for an organism to grow upon. So if it doesn't like glucose and the only sugar you place in that medium is glucose, then it's not going to be able to grow. So if you're interested in just growing something, not caring about what it eats, but just trying to grow it, then you might use complex media in order to do that. Now there's other types of media as well, and these categories of media are designated based upon the fact that they're going to tell you something about the organisms that are growing upon it. So let's start with selective media. Selective media indicates that there's some element, there's some compound within the ingredients or the recipe of this medium that is going to inhibit the growth of a particular group of organisms. And therefore, when you look at this plate, when you observe the results, you observe growth on this plate after you've inoculated it, when you see something growing there, you can actually state something about that organism, some characteristic. So for instance, in Thyre-Martin agar, you can use this to isolate a particular STD-causing bacterium known as Neisseria gonorrhoeae. So again, if you have a patient with an STD, and you want to, you collect a pap smear sample, let's say, and you want to try to figure out what exact type of STD this is, well, you could take your sample, grow it up on this Thyre-Martin agar, and if there is growth, that's pretty good evidence that the person has gonorrhea. If you don't see anything growing, then it's likely some other type of disease-causing STD, a disease-causing pathogen. Another example is MacConkey agar. This can be useful for isolating gram-negative bacteria. It basically has an antibiotic added to it that inhibits the growth of gram-positives, almost like penicillin. So therefore, you see anything on the agar, you can automatically assume or presume that these species are going to be gram-negative without actually doing the gram stain. Now another category of media is differential. And the thing about differential is that there's going to be something, in differential media you have a change that occurs, or a differential component that will again tell you something about the organism. So the bacteria that grow upon this organism will change it in a certain way, and that again gives you some information on what this organism is, or what it does. So a couple of examples here. Blood agar is shown here on this top figure. And in blood agar, when you get it, it looks blood red. It looks really dark red. And as you add organisms to it, and the colonies begin to form, you can observe or not observe a clearing of the colonies themselves. So when we look here, we see some organisms, some colonies here, in which the zone around the colonies, the areas around the colonies, have turned from that sort of dark red color to something much lighter. This is due to a process these bacteria can undergo that's known as hemolysis, which is basically them producing an enzyme that lyses red blood cells. So you see this zone of clearing around a colony. You can presume that these cells can actually synthesize a protein, hemolysin, that will destroy red blood cells. Other colonies here, you see these here, you don't see this light zone of clearing, but you see a more greenish zone of clearing. That indicates partial hemolysis of the red blood cells. And furthermore, if you see this colony right here, these smaller colonies, there's no zone of clearing at all around them whatsoever. So that indicates that while they can grow on the blood, they can grow on components of the blood, they don't actually destroy the red blood cells whatsoever. They leave them intact. So there's a lot of information that can be discerned from using blood agar. And it's particularly useful because in many cases, pathogenic strains that do lyse red blood cells, such as these, are typically going to cause more dangerous forms of disease. Here's our good friend MacConkey agar again. We already talked about MacConkey agar being selective, but MacConkey is also differential. So the terms selective and differential aren't mutually exclusive. You can have types of agar that are both differential as well as selective. So the differential component here is the fact that McConkey auger starts out being relatively dark in color, almost a purplish type of color. And when you have gram-negative organisms that grow on there, some gram-negatives love to produce acids as a result of their metabolic strategy. So as they produce acids, the pH of the plate goes down, of course, and due to the fact that there's a pH indicator molecule added to the auger, you're going to see a change in color from this darker purple to a greenish color based upon the fact that there's more acid. And this can be useful because many of the gram-negative organisms that are found in our intestines that come out in our feces, these are commonly referred to as coliforms. And coliforms traditionally are going to produce acids on McConkey, which is going to cause this color. So again, if you think a sample may be fecally contaminated, or you think that blood may have fecal contaminants in it, you could grow those samples up on McConkey auger, try to witness this change in color, which would then indicate that indeed there is some fecal contamination occurring. Now, another type of media is known as enrichment media. And the reason you use enrichment media would be when there's a certain species in a sample that you know is there, but is there in very low numbers. It's not a great competitor. So it's in the vast minority. So if you try to grow it up on normal media, the other cells, the other species in that population are always going to dominate, and you're going to have a hard time selecting for the rarer species. So it's a way in which you can set up a media type in which the rare species are given the type of environment that they can thrive in. So they're going to grow more than they normally would, and conversely, the more dominant species are not going to grow as well as they normally would. So if you take an organism, or you take a sample with a bunch of different organisms, a bunch of different species, and you know your particular species, it grows all right. I mean, it'll survive at room temperature. But if you grow it in the cold, if you throw it in the fridge, the organism you're interested in may be psychophilic. So you grow it in the cold, in the fridge, it's going to result in that particular species growing much better, and you'll be able to isolate it. There's also nutrient enrichment. If you know that a rare species can also, it also will eat organic compounds like glucose or sugars like glucose, but you also know that it'll eat fats, and maybe some organisms won't, then maybe you'll set up a medium in which only fats are available to eat, and therefore you would expect these species to do much better. So this last bullet point here, how could we enrich for media for nitrogen-fixing bacteria, which is something we'll get into in much more detail in Chapter 6, but nitrogen fixers are going to use different types of nitrogen. They're actually going to reduce nitrogen compounds like ammonia and convert them into things like nitrate. So again, if we know we want a nitrogen fixer to grow more than it normally would, we would figure out what it likes to eat, and then we would add a bunch of that to the medium.